Hello everyone, I'm Dr. Sonia Angelin, Associate Professor, Department of Life Sciences, Christy Jayanti College. In this video, we will be discussing about transduction, where the two types, generalized and specialized transduction, will be dealt with. Transduction is a phage-mediated process of genetic material transfer in bacteria. This process of genetic recombination was discovered by Zinder and Lederberg in the year 1952 in Salmonella typhimurium. During their experiments, they also wanted to discover whether the E. coli type of genetic exchange can be seen in Salmonella typhimurium. The bacteriophage acquires a portion of the bacterial DNA of the host cell in which it reproduces and then transfers this acquired DNA to another bacterial cell to which it infects. Such bacteriophage are called transducing phages. Transduction is of the following two types, which is generalized and specialized. Firstly, we will be discussing about generalized transduction. Transduction, which results in the transfer of any bacterial gene from one bacterial cell to the other, is referred to as generalized or non-specialized transduction. It is mediated using virulent phages and certain temperate phages. Examples: E. coli phage P1, Salmonella phage P22, Bacillus subtilis phages, and SP10. In generalized transduction, some of the developing progeny phages during their normal lytic cycle may accidentally acquire a piece of the bacterial DNA. Such phages, after the lysis of the host bacterial cell and their release, attach to and inject their DNA into a new recipient cell but fail to re-establish lytic cycle therein. Once inside the recipient bacterial cell, the injected DNA may be degraded by nucleases, in which case genetic exchange does not occur. The injected DNA, however, may undergo integration, resulting in homologous recombination. As a result, the transduced cell may possess new combination of genes. This is a pictorial representation showing you how a transduction takes place. As you can see, the first picture carries a bacterial cell being injected by a phage, and it's transporting its phage DNA into the bacterial cell, wherein the fragments of bacterial chromosome are being replicated in the host cell. And once the reassembly of the phages takes place, you can see that the transducing phage carries the bacterial chromosome and the normal phage carries the normal phage chromosome. When the cell lyses, the phages are released into the environment and they go and infect another recipient cell, where the bacterial DNA of one donor bacterium transported to another recipient cell where the transduction process takes place. And this transducing phage will release the bacterial DNA from the donor into the recipient cell, where the homologous recombination takes place and the DNA from the donor gets integrated into the host recipient cell, which is called as the transductant. The specialized transduction involves where the bacteriophage transfers only few restricted genes from the donor bacterium to the recipient bacteria. Specialized transduction is carried out only by temperate bacteriophage, which undergoes lysogenic cycle in the donor cell. At first, temperate bacteriophage enters into the donor bacteria and then its genome gets integrated with its host cell and the DNA and at certain locations and remains dormant and passed the generation to generation into daughter cells during cell division. The bacteriophage, which follows lysogenic cycle, is known as the temperate phage. When lysogenic cycle is exposed to certain stimulus such as chemicals or UV lights, it causes induction of the virus genome from host cell genome and begins the lytic cycle. On induction from the donor DNA, the phage genome sometimes carries a part of the bacterial DNA with it. The bacterial DNA lies on the side of the integrated phage DNA are only carried during the induction. When such bacteriophage carries a part of the donor bacterial DNA, infects the new bacteria, it can transfer the donor DNA fragments into the recipient cell. So in this specialized transduction, only those restricted genes are situated on the side of the integrated viral genome and have a chance to enter into the recipient cell. So this is a pictorial representation showing the specialized transduction where once the phage infects a bacterial cell, the phage DNA is released and it goes and integrates with the bacterial chromosome, and the prophage this deintegrates and picks up pieces of the bacterial chromosome. A prophage is formed inside the host cell itself. And once replication of the viral DNA and destruction of the bacterial DNA takes place, more copies are formed, and during the viral assembly, you can see that the bacteriophage takes up the prophages containing both the donor bacterial DNA and the bacteriophage DNA, and once the cell lyses, it releases the bacteriophages which goes and infects another cell, and from the donor bacterium the prophage is injected into the recipient cell and crossover takes place, and the stable gene transfer into the recipient cell chromosome occurs during specialized transduction. Abortive transduction is another type of transduction where the genes located at the phage DNA may be expressed and synthesis of functional gene production may be carried out in such cells. Cells carrying free DNA of the phage in their cytoplasm along with the host chromosome are called abortive transductants, and the process of transduction is called abortive transduction. It is considered that abortive generalized transduction intent is 10 to 20 times more frequent than the complete generalized transduction. Abortive transductants are diploids and can be used to carry out complementation tests that are provided the operational definition of a gene and helps determining whether different mutants are in the same gene or different genes. So this is a pictorial representation of the abortive transduction where you can see that the phage DNA remains as a separate entity along with the bacterial cell. Thank you.